So what I want to talk to you about is some of the work we've been doing on technology that we are actually still developing for, it's a nanoprobe for tracking genetic variations or chemical changes in a living cell system. Okay. So these are collaborators. I work with scientists, professors in development and cell biology, and also in the UCI Medical Center and Center for Complex Biological Systems. So what we're introducing here is a unique technology that can rapidly profile genetic expression levels within a living cell at a very, very high sensitivity down to single molecules. And we believe the capability opens up new methods for diagnosis of genetic diseases and a new analysis instrument in cell biology for tracking the fate of single cells in response to various external stimuli, such as drugs, silencing genes, or signaling molecules. And also, this would evolve into a new analytical tool that is likely to have a major impact in cell biology. So the current technology for gene expression uses DNA arrays to profile the gene expression levels. And DNA arrays typically require mRNA extracted from thousands to millions of cells for analysis. And if you average over thousands and millions of cells, what happens is that you reduce the sensitivity of the measurement, and it masks out really subtle variations in gene expression, which turns out to be very important in understanding what happens to tissues and cells. The ability to detect these very low fluctuation levels in gene expression in living cells down to single molecules is key to detecting genetic mutations that result in diseases and for studying the effects of drugs and signaling molecules. So just to give you an example here, here you have a collection of cells, and you're going to look at the gene expression or the mRNA expression due to a specific enzyme within that cell system, right? So as a function of time, you see that the gene expression in the cells increase, and then in the final state, you might have some fluctuations in gene expression from cell to cell, but if you look at the number of cells and the genetic expression, you essentially have a single modal distribution. Okay? So the average, if you take the average of this, the average accurately represents what is the entire population. What the population is doing is represented by the average. But actually, in real life, what you end up is that each cell provides fairly drastically different random expression levels going from low to high. And so you end up with a distribution that's not a single modal, but maybe bimodal or many different distributions here. And the final state looks like this. If you average all this, you'll still get the same average as what you have here, but actually it hides all the information that is going on in this system of cells. And here at the bottom, you see why these kind of random variations could actually result in various phenotypes in a particular system. Here you see that you have the number of cells in a population here, and this is the stochastic expression level, and you see that most of the cells are expressing below some threshold value, but occasionally some cells express above this level, and then that results in a cell that is different to the rest of these cells, and this cell acts as like a nucleus to actually organize the tissue. So these kind of random fluctuations are very important in organizing tissues and various biological processes that go on within us. So that's why it's important to be able to detect at a single cell level. And here you see two identical, the fingerprints from two identical twins, and clearly they are different for the same reason as what I just described. And here you have identical cats who are also different, and for these reasons that I just mentioned. And here is a distribution of cell fates in insect photoreceptor cells, which then result in the different photoreceptors that you find in the eye of that insect. So what is the technology we use to do this? Well, it's called the atomic force microscope. Phil mentioned microscopy. And the technology we are using is a technology that I developed with my team at IBM almost 25 years ago. It's called the vibrating mode AFM, which actually now happens to be the AFM of choice for profiling surfaces in nanotechnology. And the way it works is that we have a all silicon cantilever with a very fine needle. This needle at the end is only about 100 angstroms in diameter, it's about 50 atoms or so. And what it can sense its presence to a surface through the change in the mechanical properties of the probe as it approaches the surface. It essentially measures a shift in the resonance frequency of this very small structure. The structures, and then the object is scanned in order to kind of scan the topography of the surface and measure on a very small scale. And the size of this length is typically about 100 microns, the diameter of your human hair, and this length here is about 1/10 of that. And again, at the very end, you have 100 angstrom radius probe that can probe the surface. So we start with this, and of course this technology has evolved into a whole bunch of different microscopes for measuring all different types of physical properties, from force to light to heat to magnetism and charge and photo voltage and so on. But here what I want to talk to you about is how we're using this, or how we're modifying this to do this single cell probing. So about 5-6 years ago, we learned how to manipulate molecules up and down one of these probes by applying a voltage at the back of the silicon cantilever. So we can actually, by doing that, we can actually move molecules up and down the probe, and as they move down the probe, they move at different speeds, and therefore we can actually separate them out in space based on the time it takes for them to move along the surface. So that was about 4 or 5 years ago we learned to do that, and we've now taken this probe and modified it in a way that allows us to insert it into cells. So these are cells, living cells. We take the probe, we make an outer electrode, insulating film, and an inner electrode, and at the very end of this probe, then we can actually, by applying an AC voltage, we can actually create a very localized field that can be used almost like a magnet to drag molecules towards the probe. So what we do is we insert this probe under AFM control, very precise control, through the cell membrane into the cell, and we also watch it using an optical microscope from this side. And so we can target a cell, insert it, and then the next thing we do is the probe itself has little DNA strands, typically about 25 bases, attached to the very end. And these DNA strands are like are complementary to the messenger RNA you want to pick up in the cell. So what happens is that you insert the probe into the cell, you apply this field, you drag all these molecules towards this probe, and only the RNA that's complementary to these strands attach. Okay, then we pull the probe out, then we go into a system that essentially dissociates this collected RNA. We copy it into a DNA, cDNA, and then we quantify it using standard quantification techniques. So that's basically the process. And we have shown that we can use this technique then to look at, so these scans here tell you give you an idea of the sensitivity of picking up different types of RNA in a cell. So we can pick up GAPDH, which is expressed at something like 1500 molecules per cell, and this HPRT is expressed at about 5 to 10 molecules per cell. And so we essentially can selectively pick up different RNA expressions in a living cell system using this technique. We have also shown that we can look at the breast cancer gene from a single cell that has been genetically modified to express the breast cancer gene. And then we compare that here with a normal cell where you see no RNA expression from that cell. So these are actually, and then here is some data which shows that we can actually watch a cell as it goes through apoptosis, which is as it dies. We can actually map the microRNA as a function of time as the cell goes through cell death. So for the first time, then we can actually monitor what happens in the chemistry in a cell as a function of time. So what can you do with this? So I think there are lots of applications, and I just show you 2 applications because of the lack of time. So one application here is to look at these epigenetic changes that contribute to various diseases, such as cancer and other diseases. And so if you know your DNA is wrapped up inside your nucleus in this way, and you have methyl groups that attach onto different sites on this DNA, and when these methyl groups are attached, they can switch off gene expression in that local region. And so the methylation patterns in your DNA then is very important in terms of, because it describes which genes are on and which genes are off, right? So when you don't have any methylation, basically those genes come on. If you look at a group of normal cells, you can basically develop cancer through different pathways. Here you can develop it through a DNA mutation, which basically then causes cancer and which proliferates, but you can also develop it through these epigenetic changes. So these methylation patterns, changes in them, can actually create primed cells that eventually can result in DNA alteration and then a cancer here, which then goes here. Or you can have an epigenetic change which directly causes a change right immediately. And what we're trying to do then is to actually go into a single cell and measure the epigenetic, the RNA expression from the paternal and maternal part of your genetic code, and look at those changes. And by looking at those changes, we can then know whether that person has a specific disease or is likely to get a specific disease. So these are data that we just got from using this technique, again single cell, and we extract the RNA from that cell, and then we look for different types of RNA expression, and we can see these allele-specific expressions from a single cell. So this is right at the beginning. We haven't, we need to do much more work to actually correlate that with, various disease states and so on. The other area that we are working on is to use it to look at the development of fertilized embryo. And as you probably know, when you have an egg that's fertilized, the patterning of this egg into structure is dependent on the RNA that is injected from the mother into the egg when it's laid, and that gradient of RNA then creates the banding and patterning that then results in further structure in the egg. We are able now to go in with these probes and measure these gradients. So these are measurements taken at different points along this fertilized egg, and we basically can see the gradient of RNA along this fertilized egg. So we can compare this technique with other emerging technologies. I'm comparing with microfluidics. So I think one of the key things is that we can do this with living systems. We can follow what goes on as a function of time. We can possibly use it to detect diseases. It's very, very fast. It's reasonably simple. Right now it's not simple, but we want to make it simple. There are no chemical, the very important thing is that there are no chemical purification steps to do this. You just insert the probe into the cell, you extract the RNA, and then you measure it, right? Here you have to lyse the cell and kind of digest all the unwanted material before you can do your assay. And in principle, you can measure proteins, detect proteins with this as well. And we are in the process of doing some experiments where we want to inject material into the cell. And also we can look at live cell response. So eventually where we want to be is to do this. So you have a cell under a microscope, target it, do the sample, and then put it into a little cuvette, and then essentially do repeated measurements as a function of time into a series of cuvettes, and then go and quantify it. So that's basically what I wanted to talk to you about today.